Okay. Hello, everyone. I'm excited to share with you the work developed during the sPACE HackWeek 2026 or 25. Our project is a glimpse of food webs from space. We worked together with Josefina and Patrick, and our mentor was Jonathan. And the central idea is simple but powerful. Phytoplankton from the base of the marine food web, their composition and abundance can influence the structure of fish communities through the food web change, and by integrating satellite-derived phytoplankton data with troll surveys, we aim to uncover how these tiny organisms cascade through trophic levels to shape these fish assemblages. But we stumbled upon the fact that there are limited spatiotemporal overlap for zooplankton and moana products, so we adjusted the matter and we excluded these intermediate trophic levels to focus on the direct relationships between ocean color and fish functional traits, because in the first place we expected that distinct phytoplankton functional types determined zooplankton size and larval habitat suitability, cascading to support specific fish gills. So our workflow included taking the fish data from the NOAA bottom trawl surveys, the phytoplankton data from PACE satellite products, including Moana, chlorophyll A, and also Modis aqua sea surface temperature, and the fish functional traits from the fish-based dataset. So we constructed 3 matrices, one with the environmental information, one with the species traits, and one with the composition and abundance of species in each station. And by doing this, we can run an RQL analysis that basically finds the main gradients that connect the environment, species, and traits simultaneously. Essentially, it shows which environmental conditions favor particular traits, and by extension, which species are associated with those conditions. So this allows us to see how changes in ocean color and temperature across seasons affect the functional structure of fish communities. So for the practical purposes of the HackWeek task, we focused on the Northeast US shelf, spanning from 34 to 40 latitudes and longitudes from 75 to 71, and analyze the spring and the fall data. To standardize our analysis, we identified the top 10 most abundant species, capturing over the 19% of the total catch in spring and fall, and we curated their functional traits such as trophic level, maximum size, diet breadth, and mouth position. And we chose these traits because they were the ones we believe that gives us an information about the particular question that we have. And about these images you see here, we have a quick look at the raw satellite data and environmental data we work with. This is for fall and for spring, and these maps shows the spatial distribution of several PACE product that we use, like the chlorophyll A and different phytoplankton groups, as well as the Modis SST. And the goal here is not to interpret each map in detail, but rather to give you a sense of the scale, the coverage, and the resolution of the data that we were integrating with the fish surveys.
Okay, so for the results, I will probably talk about the spring results, just because I don't want to go over time, but we got pretty similar patterns. So this is what the RQ looks like. So here, as you can see, the x-axis of our B-plot is capturing most of the data variability, so we can focus on this horizontal gradient. And what this figure tells us is that to the left, we're going to have bigger fish with higher trophic levels that are specialists because they have the lowest diet breath. And these traits were associated with bigger plankton, lower sea surface temperature, and higher bottom temperature. And then on the other side of the figure to the right of your screen, we have smaller fish with lower trophic level who are generalists. And this was associated with smaller plankton, higher sea surface temperature, and lower bottom temperature. Next slide, please, Maite. And then, like I said, for fall, we had practically the same. The only difference was that body size was not that much of a predictor for the species positioning within the figure, but the pattern was similar. We can move on. So the main takeaways from this were that the phytoplankton community seems to be structuring the trophic and functional composition of fish, that there's a link between primary producer size and the dominance of either a generalist or specialist fish, and that PACE can provide much richer resolution that allows us to see this functional base of the marine ecosystem. Next one, please. So we thought, what could be driving these results? And since bottom temperature and sea surface temperature seem to having an opposite pattern, we were thinking maybe there's some stratification that is causing this. So in stratified stations, maybe nutrient flux is not as efficient as in more homogeneous water, in a more homogeneous water column. So since there's less nutrient input for the euphotic zone, plankton may be smaller, and then in more homogeneous places, plankton may be bigger. This would lead, according to us, to longer food webs in the more stratified areas, because since phytoplankton is smaller, you can have a wider variety of zooplankton feeding on it. And then, so since the food web is longer, there's more energy loss till we reach the fish. So maybe that's why fish are smaller and more generalists than in the other case. Next one. Okay, and so if we had more time and what we would like to explore later, we would like to have some information on zooplankton to put to test our hypotheses. We would also like to incorporate some traits that give us an idea of growth rates of the fish. This is especially useful for fisheries management because fast-growing fish are less vulnerable to fishing. So maybe if we could link growth to certain environmental variables, that would be useful. And then to expand on the time series of the analysis. This year we only have the 2024 data to work with, but as one of the other groups said, as the years go by, we will probably be able to extend this. Next one. Okay, so we wanted to talk about some of the exciting things we learned to work and extract PACE data. It was also our first time working collaboratively in GitHub. It was really useful. And we managed to get the best of both worlds of Python and R. So it was really great. Going back to Eli's first figure showing how it would be easy to learn. Our journey was quite a roller coaster, but we had a really, really nice time. So thank you, everyone.